Today I am taking up part 1 of sexual reproduction in the plants. I am dealing with microsporogenesis and megasporogenesis. That is formation of pollen grains and formation of ovules. Here I have drawn a longitudinal section of a bisexual flower. It shows all the components of a flower. The basal part of the flower is known as a receptacle or thalamus. It is a platform-like structure. On the thalamus, we find all the floral parts. The floral parts constitute 4 whorls. The outermost whorl forms the calyx and it is made of sepals. After the calyx, there is the second whorl made of petals and known as corolla. If both petals and sepals are identical, as in some plants, they are together known as perianth. Then, sepals are usually smaller and greenish in color, whereas petals are bigger in size, in different colors. They are scented and they are also highly attractive. After the petals, we find stamens forming the third whorl. Stamens may be many, and each stamen has 2 parts. This is the anther and this is the filament. In the anther, pollen grains are formed, and all anthers are bilobed and dithecous. Bilobed means the anther has 2 lobes, and each lobe has 2 theca. The innermost whorl is made of the gynoecium. Gynoecium consists of a number of carpels. A carpel is made of 3 parts. This stigma, where the pollen grains are received. This is the slender style which connects the ovary with the stigma, and this is the ovary which contains ovules. Below the stamens, we come across nectaries in insect pollinating flowers. These nectaries produce nectar, which is a sugary solution. It attracts the pollinators. This is the structure, and here also one more part. There is a bract also. Below the flower, there is a structure called leaf-like structure called the bract. Now let us take up the formation of pollen grains. Now, formation of pollen grains is known as microsporogenesis. Microsporogenesis. This is the figure of a mature anther. A mature anther, I have told you, is a bilobed structure having 4 theca. This is 1 theca, the second one, third one, fourth one. Each theca consists of a pollen sac. This is the pollen sac. Inside the pollen sac, there are a number of cells known as sporanginous tissue. Then, the pollen sacs are surrounded by 4 membranes or layers. The outermost layer is known as epidermis. The second layer is known as endothecium. The third layer is known as the middle layer, and the fourth one is known as tapetum. Then, in the pollen sac, pollen grains are formed from sporanginous tissue. And this part is called line of dehiscence. When the pollen grains are formed and ready to be released, at the line of dehiscence, the anthers split open and the pollen grains are released to the outside. Now let us take up microsporogenesis, that is formation of pollen grains. Because of their smaller size, is known as microsporogenesis. In the sporanginous tissue, I have told you there are a number of cells. And these cells develop into pollen mother cells. The pollen mother cells have diploid number of chromosomes, that is 2n number. The pollen mother cells undergo first meiotic division and produce 2 daughter cells. These daughter cells have only haploid number of chromosomes, that is the chromosomal number is reduced to half. Then these 2 cells undergo second meiotic division and produce 4 cells. These four cells are also haploid. You know that the second meiotic division is nothing but a type of mitotic division. Initially, the four cells are attached together and they are known as a tetrad. Afterwards, due to drying up, the four cells get separated and they are known as the free pollen grains. If you observe a pollen grain under a microscope, it looks like this. It is covered by two walls. This is the outer wall, this is the inner wall. The outer wall is known as exine, inner wall is known as intine. The exine is very thick and also very strong. This exine is made of a substance known as sporopollenin. Sporopollenin is a highly resistant substance to temperature, acids, alkalis, and also for enzymes. Till now, no enzyme is known that can digest the sporopollenin. Because of this hard nature of the sporopollenin, the pollen grains can be preserved in the soil for many millions of years. Then the intine is made of cellulose and other substances. Then the actual pollen grain is surrounded by a plasma membrane. The cell is surrounded by plasma membrane. Then, at some places, the exine leaves some small apertures. They are known as the pits, through which the pollen grain germinates and the pollen tube comes out. Then, inside the pollen grain, I told you initially there is only one nucleus, but the nucleus divides and two cells are formed. Out of them, one cell is bigger and one cell is smaller. The bigger cell is known as the vegetative cell. It contains a large amount of cytoplasm laden with food materials. The smaller cell is oval in shape and it floats in the cytoplasm of the vegetative cell. And the smaller cell is known as germinative cell. In 40% of the plant species, the germinative cell divides again and forms two cells. It means the cells are three, or the pollen grain is at three cell stage. So if three cells are formed, it is known as three cell stage. But in 60% of the plant species, only two cells remain in the pollen grain before it falls upon the stigma, and it is known as two cell stage. In this way, the pollen grains are either in two cell stage or three cell stage. The pollen grains are also sometimes harmful to human beings and other animals. Some pollen grains cause asthma, bronchitis. These are one type of allergies. One famous example about the pollen grain causing allergy is the Parthenium plant. Parthenium plant is not our native plant. It is exogenous plant imported to India unintentionally through wheat. When we imported wheat from America under PL 460 scheme around 1960, these Parthenium seeds also entered our country, and as the climate is suitable, they got established. Now they have become endemic and they are causing a lot of problems. The pollen grains are allergic not only to human beings but also to a number of animals. Then, nowadays, the pollen grains are also consumed by human beings, thinking that they enhance the performance of sportsmen and also the performance of the racing horses. Just like the seeds, the pollen grains can also be stored for posterity at minus 196 degrees centigrade. At this temperature, they can be stored for many years or even centuries. Then, the lifespan of a pollen grain is very short in wheat and rice, only 30 minutes. In families belonging to Rosaceae, Leguminaceae, etc., the pollen grains can live for many years. In this way, pollen grains are formed through microsporogenesis. Now let us take up megasporogenesis. Megasporogenesis means formation of ovules. In the center of the ovisac, we find cells constituting nucellus. Nucellus is made of a number of cells. This nucellus is surrounded by two integuments. This outer integument, this is the inner integument. These two integuments surround the nucellus, and at one point they leave a small opening. These two integuments leave a small opening. That opening is known as the micropyle. The end of the ovule at the micropyle is known as the micropolar end, and its opposite end is known as salazal end. Then the ovule is attached to the placenta through hilum and funicle. Then one of the cells present in the nucellus develops into a megaspore mother cell or embryo sac mother cell. Though there are a number of cells in the nucellus, one of the cell near the micropolar end here develops into megaspore mother cell or embryo sac mother cell. This cell has diploid number of chromosomes, that is 2n number of chromosomes. This megaspore mother cell grows in size, and after reaching maximum size, it undergoes meiotic division. Through meiosis 1, it produces two daughter cells, which are haploid. These two daughter cells undergo second meiotic division and produce four cells. These four cells are known as megaspores. Out of these four megaspores, three megaspores disappear or disintegrate, and only one remains. The remaining one is known as functional megaspore. Thus, a difference exists between microsporogenesis and megasporogenesis. In the case of microsporogenesis, what is happening? All the four pollen grains are alive. They are functional. But here, three megaspores are disintegrating and only one remaining. And this functional megaspore undergoes nuclear divisions, which are all mitotic divisions only. The nucleus of the functional megaspore undergoes first meiotic division and produces two daughter nuclei. Each daughter nuclei moves to the opposite poles, undergoes second division, forming two daughter nuclei at each end. Then these two daughter nuclei divide again, and at the end, four daughter nuclei are formed at each end. In this way, altogether, 8 daughter nuclei are formed. The interesting thing here is the nuclei are dividing, but the cytoplasm is not dividing. That's why these are known as free nuclear divisions. After some time, 6 nuclei are surrounded by cell membranes and 2 remains free. The free nuclei are known as the polar nuclei, and they remain in the center of the cell below the egg apparatus, and now the cell is known as central cell. So the polar nuclei remain in the central cell. 3 cells form antipodals at one end, and other 3 cells form egg apparatus at the opposite end. So there are 3 antipodal cells. This end is known as salazal end. The egg apparatus is formed at the micropolar end. Out of these 3 cells, one cell forms the egg cell, future zygote, and on either side of the future zygote we find synergids. Synergids means associated cells. These synergids have filiform apparatus. This is a red colored structure. The filiform apparatus helps in guiding the pollen tube during fertilization. In this way, the ovules are formed. This is the developed ovule or embryo sac. Then, there are some terms you have to remember. Monocarpellary. If there is a single pistil in a flowering plant, it is known as monocarpellary. If there are more than one pistil, it is known as multicarpellary. If the pistils are fused, they are known as syncarpous flowers. If the pistils are free, the flowers are known as apocarpous. Then, in the case of wheat, paddy, etc., the ovaries contain a single ovule. But in the case of papaya, watermelon, orchids, etc., the ovarian cavity may contain more ovules. So in this way, a megaspore develops from the cells of nucellus. So this is about microsporogenesis and megasporogenesis. In the next class, I will take up pollination in plants. Thank you.